Well, in time, so now we can open the discussion of the challenge, and some of the presenters, both from the organizers or the participants, had some questions or comments that we could discuss now, or I don't know if we should start with those or just let people ask directly, but so maybe we can now, people can ask questions as normal pertaining any of the aspects of the challenge, from the presentation, from the organizers to the specific teams. Well, I'll start maybe first, if no one wants to break the ice. So to the point that you raised about the effect of when you have the drug on a protein, how this makes an increase in the level. And well, Laura can maybe comment as well, but I was thinking if it could be another phenomenon, which is that the drug is essentially affecting the structure of the protein, and it could open the structure so that, for example, the phosphorylation site is more accessible, and you have a hyperphosphorylation, which doesn't necessarily mean higher activity. That was how I was thinking, but maybe Laura can comment on this.
The protein actually is phosphorylated, but it's in a configuration where it's no longer active. So it's, at first glance, a very counterintuitive observation. But this, I think, has been described. And I'm guessing if we see it here in this particular case, we probably see it elsewhere. So it is something that is important to think about, that at first blush, the problem is, as we have observed time and time again, more complicated always than we think it should be.
Josh?
Isn't this a classic case of feedback in a biological system? You inhibit at one level, and the cell responds to rescue that protein whatever way it can, and it just upregulates it. Or am I misunderstanding?
No. I think that it's true that also is happening, but I think this particular case has been described as a conformational change.
Also because it happened very fast, right? So.
Yeah, that was the piece that we were sort of struggling with in our hypothesis and thinking about it, was that it was happening so quickly.
Fast.
We didn't understand how you could see that immediate increase. So thanks, that's helpful.
Okay. So, Gustavo.
Yeah, so one question for the experimentalists. How much, when I see these RPPA arrays, and I wonder how well characterized is the noise? Are we dealing with how much is signal and how much is noise? And to some extent, I don't think that we address that completely during the challenge.
I think this is something that we don't have a really good sense of. And I think this is true not just in this particular data set, but as a community coming from trained in biology. I think this is something that we're not always appreciating. And it is this sort of exercise of making sure that we're collecting enough data, enough replicates to really be able to assess that. In this particular case, what we decided to do was, rather than make true replicates, other than as has been discussed in these talks, there were a lot of replicates at time 0. And rather than do a lot of true replicates, what we decided to do was really search the space deeply with the idea that repeating along some dimensions would help us get at this idea of how much noise might be in the system. But it is an important consideration. And something too to remember, that all of these sorts of experiments are really screens. And the idea is that we're going to be learning about the biology, which will then be further followed up with additional experiments.
This is kind of fun. I feel like I'm on Phil Donahue or something. How different, without giving away for the next, for these follow-up challenges, how different are the networks you're getting from the 32 different conditions and when you guys look at them, are they, the fact that the prior does so well makes me feel like it's not that different, but maybe Maybe you can comment on that.
So
This I will let Stephen comment on in Phil Donahue style. We'll just keep walking around.
So are you asking about the submissions from participants or the real networks? Well, in reality, there is no real whole network structure, right? There's just this list of descendants. But yeah, there is a significant overlap between the different cell line and stimulus regimes. So, for example, when you apply a certain inhibitor, you're likely to see a certain change in the same proteins across all the stimuli, if that inhibitor really has a clear effect. For example, things downstream of an AKT inhibitor, P70 will go down into all the stimuli. So in that sense, the prior information, it makes sense that it plays a big part. So yeah, I would say they're quite similar, but I think the thing that can be added by the kind of machine learning type of methods is the things that are less expected to add on to the prior knowledge.
So dear conference organizer and attendees, I represent the team at MSU Sun Lab. My student Yang and I are happy to be here today at the most participated DREAM challenges in history. I congratulate organizers and participants for their enormous contributions to system biology research. On the other hand, I, my student, and my institution, New Mexico State University, are disappointed that DREAM 8 organizing committee chose not to make the most consistent method across sub-challenge 1A and 1B, the best performer of the sub-challenge, without clear reasoning. And also the chair informed our team that we will not be co-authors on the overview manuscript to Nature Methods. Among the top 7 teams in 1A, our method is the only one without using any prior knowledge, and the performance is statistically very close to the best in 1A as judged by the median z-score and the associated p-values and the q-values. Nevertheless, we are excited about this opportunity of sharing our work with the community, and we're also trying to learn from other outstanding teams. We thank the current and past DREAM organizing committees for creating such a wonderful opportunity that we celebrate. You are welcome to our poster number DREAM 9. Thank you for the attention.
Thank you, Joe. So the issue that Joe is raising is an important one, which is who wins, who doesn't win. And I would like to make the point that there are two levels of winning these competitions. One is what we win collectively as learning about an important problem, and the other one is what individual teams learn, and that doesn't necessarily optimize the same cost function. So for us, the issue of us learning from each other is more important than who is deemed the best performer. But for each team, who is the best performer is probably the most important thing. So I don't claim to have the absolute right way of reasoning, but this is something that we should discuss later. And I think that I don't want to not acknowledge that there was an elephant in the room there, that someone feels that there is an elephant in the room and has to be acknowledged. The last thing I want to point out with respect to the biology of it is that on the one hand, and related to the question that Joe asked, is the fact that when we looked at it, what won to some extent, and Artem, you presented that in your presentation, is the prior knowledge. That was what contained probably most of the information. Not unlike previous challenges, for example, the breast cancer prognosis challenge, in which we saw that the clinical information contained the bulk of the information rather than genomics information that was incremental to some extent. So I wonder whether there is something to be happy about that, and to some extent. And on the other hand is, what else should we look into? What other data should we look into in order to make the best of new information that makes us go beyond the prior knowledge? And to that extent, I wonder whether single cell information would actually unravel things that we cannot see from whole subpopulation lysates. Anybody wants to address that?
You were looking at me the whole time, so I assume it's a question for me. I think so it's a two-part question really. One is this issue of the biological prior and how much are we recapitulating those findings. And it is always good when you do some studies and at least some of what you find confirms what you already know. Because if everything's novel, that means something's probably wrong. I think the key to interpreting the results here is going to be looking at where we don't see that agreement. So we see enough agreement that would suggest to us that we have done an experiment where we think we're confident in what sorts of results come out. But then the question is, where do we, what are the broken links that don't match with the prior knowledge? And how are the other thing that we haven't actually really discussed at all here is, how are these individual cell lines broken in different ways? Because we think that, and we know that they have very different mutational spectra. For instance, one of them is a HER2 amplified cell line. We know that's a very important clinical feature in breast cancer. And so we expect that cell line to have slightly different signaling around HER2 than the other cell lines might. So that I think is how we would gain insight in thinking about translating the results forward. But this other question, the other issue that you raised about single cell measurements, I think also is a very important one. And this idea is something we have started in the lab to really appreciate, this different level of heterogeneity. So when I talked about heterogeneity, it was across a population of patient samples. But in fact, if we look within an individual patient, we see that tumor is very heterogeneous and that it's composed of cancer cells that themselves may be doing slightly different things. And what we're measuring in a lot of these experiments is simply the bulk activity of a population of cells, and maybe looking at individual cells will help to shed light on what's really going on.
I just wanted to respond to that. I think it's a really heartfelt statement that we heard. And, I guess we have to remind ourselves what we're really after, and who this is for. This is for patients and getting the best network for predicting what's going to happen in a cancer cell. And there's a combination we learned from this challenge. One is we can't keep making the same mistakes as a bioinformatics community, starting from scratch, just data mining. We've learned a lot, and I think the prior shows us that. And it happened, that's why I like these kind of challenges, because it's not the first, right? CASP, maybe the first kind of open competition that started this, and what did they learn from CASP in the very beginning? Alexei Merzin beat the hell out of everybody because he knew more about protein structure. And he could beat all the algorithms, and you turn on the algorithms and they move away from the native structure. When we did the, I can't remember what Steve Brenner's is called, KG, right? The critical assessment of genome information. The team that won the first time was Rachel Karchin's group, and what did they do? They went through the literature, they read a lot of papers. Humans beat the computers, right? So we're still at the beginning, this tells me, and we all love to write fancy algorithms, and we should, and we want to get to that point where the insights are going to be the real key things that move us a step forward. But we have to start at the right platform, and I'm sorry we had to learn it again this time, that the prior, damn it, is the one that's beating everybody. But maybe in the subsequent challenge that we have, in the follow-up, we'll see it's a combination of the two teams, right? Start with the prior, and maybe you guys whip the pants off Granger causality. I hope you do. So I think it's a great experiment you guys ran, and we learned a lot.
Thank you. Actually, and one quick follow-up, which is this idea of this next bonus round where people are really coming together and sharing their code, really getting into the details of what's going on. I think will give us the chance to really learn even more than what we have so far. And I think we hopefully will get to the best solution possible. But if we're all now on this one platform, I think we have the possibility to do that. So I hope that people do engage in this next round. Do we have time for one more comment?
Hi, this is Yang Xie from University of Texas Southwestern Medical Center. First of all, I think Gustavo and all the organizers did really great job to organize this Dream Challenge, and I really feel this is really one of the best platform to collect the most innovative idea from the community, and it can really contribute to the research and patient care. But I think the previous people's comment is also kind of important to at least to start to think more and for the future organization, because I think first of all, it is very hard to make an the most fair system that every participant will be happy. It is very hard. But on the other hand, I think it is also very important from the organizers and everyone to make the system be very open, and also let the participants feel that they will get enough kind of the outcome from this participation. Otherwise, I think probably the people will get discouraged from the participation. So in terms of that, I think, for example, a very clear specification of what is the evaluation criteria, and also what is the reward. For example, I was a little bit surprised that they will not be the part of the co-author of the Nature Method paper. I think this is also kind of need to be discussed very openly with all the participants, so that moving forward, I think as a whole community, people will be really want to participate and have really good motivation. I think we want to achieve both goals. First, we want to say that, okay, as a community, the crowdsourcing will be the best. But on the other hand, we also want to solicit the most talented people in the field to participate in this challenge. Okay, this is just my stuff. Thank you very much.
Thanks. Well, I think at the end of today, there will be a discussion where we really want to discuss all these things in the context of different challenges. I don't know if you want to comment anything or...
Julio, before you wrap up the session,
Yeah.
just I want to say that it's not quite accurate that that team NMSU Song Lab is not going to be part of the Nature Methods paper. We are still evolving the thinking on that. They are going to be part of the DREAM Consortium and appear, all the names of the participants will be part of that paper. And we are still thinking what to do with the in silico. I mean, we are discussing internally, but I hope that we will discuss also later whether we can write two papers. If Nature Methods would accept two papers, one in silico and one compared to experimental and one experimental, that would be the best case. The problem is that it seems that we have so much in this challenge that if we stuff everything in one paper, it's going to be impossible to read. So that's the complex cost function that we're trying to optimize. We'll discuss more about this later, but why don't we wrap up?
Yeah, with that final word, let's finish the session thanking again all the speakers, but also all the participants who made this possible. And yeah, see you in a bit. Thank you very much.